Hello students, welcome to Saurasya classes. Today our topic is biotechnology and now we are going to discuss about the genetic engineering and its importance. So now at first, what is genetic engineering? An engineer fixes a machine to make it work efficiently. Body is like a machine and genes. Body is like a machine and genes, the nucleotide sequences in DNA have the information for products to run this machine. So, the nucleotides, the sorry, the nucleotides tide sequences in DNA have the information information for products to run this machine, that is our body, that is human body. With progress in molecular biology, with progress in molecular biology, techniques have been developed by which a scientist can now manipulate genetic material. A scientist can now manipulate genetic material replace genes or replace gene products in the body. Replace genes or gene products in the body. Make identical copies. Through this replacement, they can make identical copies of these genes and store them and it stored them in a gene library, This is called genetic engineering, and this is called genetic engineering. So, in brief, the nucleotide sequences in DNA have the information for the products to run our body, and the process in molecular biology Techniques have been developed by which a scientist can now manipulate genetic materials and replace genes and genetic products through that manipulation of genetic materials in our body. And then a scientist can make identical copies of these genes and store them in a gene library, and this process is called genetic engineering. And now we are going to discuss the importance of genetic engineering. The importance of genetic engineering. We all know that diabetes mellitus is a genetic disorder. Diabetes mellitus. This is a genetic disorder. A diabetic patient lacks a gene which has the information for synthesis of insulin. A diabetic patient lacks a gene which has the information for synthesis of insulin. Synthesis of insulin. And because of this, diabetes occurs. So, such a person cannot secrete insulin. So, such a person cannot secrete insulin. Sorry. Now, take another example, such as a person suffering from thalassemia lacks of in thalassemia a person lacks of the gene for hemoglobin, lacks of the gene of hemoglobin. And can survive only through frequent blood transfusions. A person suffering from sickle cell anemia has an altered gene whose product makes the red blood capsules abnormal, and then exposure to oxygen because they contain faulty hemoglobin. Now, humans suffering from genetic disorders such as those cited above have now hope in genetic engineering, because genetically engineered copies engineered copies genetically engineered copies can help in production and storage storage in gene library of DNA. So it can help the production and storage of DNA in the gene library to be used when required. Now, we know about the use of microbes to produce various products on a commercial scale. Currently, bacteria are being genetically manipulated to act as biological factories to produce various kinds of proteins. So, with the use of biotechnology, bacteria are being genetically manipulated to act as biological factories to produce various kinds of proteins such as enzymes, hormones, antibodies, through genetic engineering. So, with the help of genetic engineering, it is possible to use the bacteria as the biological factories to produce various kinds of proteins such as enzymes, hormones, antibodies, etc. Now, the researchers have isolated genes which can be used to produce effective vaccines. Now, the researchers have isolated genes which can be used to produce effective vaccines. Researchers have also developed bacterial strains. They have also developed bacterial strains through genetic manipulation which can degrade harmful environmental pollutants, which can degrade harmful environmental pollutants. So these are the main importance of genetic engineering.